I'm Xingshun Xu from the Department of Neurology, the Physiology Hospital of Nanjing Medical University at Suzhou City. Today, I'm glad to talk about the therapeutic potential of targeting the Nr2e3 Tet2 pathway for depression. I'm glad to share our findings in my laboratory. As you know, epigenetic modifications play a critical and complex role in the mechanism of depression. Many epigenetic modification like DNA methylation, histone modification that can affect gene expression without changing DNA sequence. This has opened up a new venue for the understanding and treat depression. Among different DNA epigenetic modification, DNA methylation stand out as a key epigenetic modification. Recently, most of the studies have shown the changes of DNA methylation patterns affect many gene expression that relate to mood regulation and stress response, which contribute to the development of depression. Although DNA methylation play a critical role in the depression, similarly, demethylation also have an important role in the pathophysiology of depression. Tet enzyme mediated demethylation are at the heart of demethylation. This Tet enzyme can convert methyl cytosine to hydroxy methyl cytosine and initiate a series of reactions that can lead to demethylation. This process is critical in reactivating gene expression and maintaining cell functions. Tet family comprises 3 members, including Tet1, Tet2, Tet3. These Tet enzymes has different biological functions, including embryonic development and neuronal differentiation. Previous studies have shown dysregulation of this enzyme can cause multiple diseases, including cancer and neurological diseases such as dementia. Due to the distinct structure of Tet enzyme and their cell-specific and tissue-specific distribution, they have different function. For example, previous study have examined stress-induced phenotypes in Tet1 knockout mice and Tet2 knockout mice. Our findings indicate that the Tet1 mice exhibited resistance to stress. However, in Tet2 knockout mice, these mice became highly susceptible to stress. Stress increased immobility time in forced sweet swimming test and tail suspension test. This highlights the unique role of Tet2 in stress resilience. This is distinguished from Tet1. Interestingly, stress can significantly increase the expression of Tet2. This implies compensatory adaptation to restore Tet2 to maintain the homeostasis balance. Conversely, Tet2 knockout can cause depressive behavior. So this suggests Tet2 have a protective effect to stress. We have known Tet2 enzyme activity is regulated by different transcriptional factors as shown this figure. But how Tet2 expression is controlled is remained unknown, so we examined the regulation of Tet2 expression. By using different three complementary bioinformatic tools, we identify the potential transfection factor for Tet1. By intersecting the result from the analysis, we finally identify 119 transcriptional factor as a putative candidate for Tet2 expression. Through comprehensive screening of transcriptional factor based on binding motif score of Tet2 and functional validation, we identified 10 candidate transfector factor. After subsequent experimental validation in the hippocampus of stressed mice, Nr2e3 has emerged as a promising candidate for Tet2 expression. Previous studies have shown Nr2e3 is a master transcription factor in regulating photoreceptor cell differentiation, such as the information of cold cells and rod cells. But the potential role of Nr2e3 in modulate Tet2 expression remained unexplored. So we conducted functional validation studies. First, we examined Nr2e3 expression in stressed mice. We find Nr2e3 significantly increased in the brain. This indicates Nr2e3 is involved in the hippocampal stress response and contribute to the behavior and molecular change. By using loss of function and gain function, we found that Nr2e3 knockdown can significantly decrease Tet2 expression. However, Nr2e3 overexpression markedly increases the Tet2 expression. This indicates Nr2e3 is a positive regulator of Tet2 expression. To further identify the causal relationship between Nr2e3 and Tet2, we construct Nr2e3 knockout cells in Tet2 cells. Our rescue experiment demonstrated that Nr2e3 reintroduction restored the Tet2 protein to basal level. This result indicate Nr2e3 directly control the Tet2. To further determine how Nr2e3 control Tet2 expression, we constructed a different Tet2 promoter plasmid. By using this plasmid, our experiment demonstrates Nr2e3 directly bound to the Tet2 promoter, specifically to a region known as motif 10.3. But interacting this motif, Nr2e3 can promote the Tet2 promoter activity and lead to the Tet2 expression. We use adenovirus to knock down Nr2e3 in the hippocampus of mice. We inject the adenovirus to bilateral hippocampus. Our finding, we indicate Nr2e3 and the Tet2 significantly decreased in the mice. This in vitro study demonstrate that Nr2e3 is a critical regulator of Tet2. But the Nr2e3 reduction in the brain may cause the downstream effect of Tet2 dependent process in brain. As speculated, Nr2e3 knockdown causes depressive behavior in mice. Nr2e3 knockdown increased the immobility time in test tail suspension test and forced swimming test. Also, changed the social behavior in these mice. This indicates Nr2e3 have an important role in maintaining normal behavior and have a potential role in the depression pathogenesis. Further, we examine the changes of DNA 5-HMC modification. We find that the change DNA 5-HMC modification is in the various genomic region. Specifically, these are related to some genes that relate to mood regulation and synaptic function. So we first further examine the protein expression, such as PSD95, NMDA receptor 1, synapsin 1, in the Nr2e3 knockdown mouse. We found that this synaptic protein decreased in the brain. This indicates the dysregulation of this synaptic protein may contribute to the pathophysiology of depression. Considering the critical role of Nr2e3 and Tet2 pathway, so we developed a cell-based platform to screen drugs that can enhance the Nr2e3 Tet2 pathway. So candidate compounds were evaluated for the ability to increase the expression Nr2e3 and Tet2 in cell models and animal models. Upon the compounds, we find azacitidine is a promising drug to increase the Nr2e3 Tet2 signal pathway. We find Nr2e3 and Tet2 are significantly increased in the normal cells, but not in Nr2e3 knockout mice. Also, azacitidine also increases the expression of Nr2e3 and Tet2 in the hippocampus of mice. More interestingly, azacitidine also improves the depressive behavior in stressed mice. Azacitidine treatment decreases immobility time in tail suspension test and forced swimming test. But azacitidine have no effect in Tet2 knockout mice. This clearly indicates the antidepressant effect of azacitidine is through Nr2e3 Tet2 pathway. So our ongoing research continues to focus on screening and validating drugs targeting Nr2e3 Tet2. So we are confident this approach can find more novel and effective compound to treat depression. Finally, I express my gratitude to my laboratory members and collaborators. Most work was done by Dr. Ma and Dr. Zhang. I have a long-term collaboration with Jiming Kong at the University of Manitoba and Dr. Xingmin Li at the University of Albert. This study was supported by grant from the Ministry of Science and Technology of China, as well as National Natural Science Foundation of China. Thanks for your attention.